But I wanted to touch on the topic of adaptive designs. This is something that is of much interest for folks that are doing randomized controlled trials because it's very appealing to be able to not necessarily have a protocol set in stone at the beginning and not be able to change things and be flexible. But that's a misconception. I'll talk to you about what are adaptive designs and how they are different from designs where you are changing the protocol. Adaptive designs are designs that are at the beginning have a protocol that is set in stone as to how you're going to do the adaptation. So we'll go so slowly through this and hopefully convince you of the difference between flexible protocol changes versus adaptive designs. So what are adaptive designs? They are, as I said, adaptive by design. They're not ad hoc adaptive, where I'm sort of changing things as problems appear. And what the problem with the adaptive design name is that it covers a lot of different possible adaptations. So you could adapt the sample size, and that is called sample size re-estimation. You could adapt who you include in your study. You may do what's called adaptive enrichment of certain subgroups of patients in your study. Or you may adapt how you randomize. So we do have adaptive allocation. You may also, and we already talked about this, do adaptive interventions. So you are changing the intervention using the platform umbrella trials that Ananta talked about. Or you could be doing adaptive starting. When I start the study, I'm going to start it earlier and then do this, and we call those the vanguard phase. We'll talk about vanguard phase. Different from pilot studies, but it's called the vanguard phase of a trial. And then we have adaptive stopping of the trial. That is the interim analysis we talked about yesterday. So almost all studies that have an adaptive aspect to them, because almost all studies have a data safety monitoring board that could terminate the study early. And so that's why in the DSMB charter, you specify the statistical analysis plan for interim analysis. They're including the adaptive stopping of the study. Okay. So as you see, there's multiple things you can adapt. You can adapt not only one at a time, you can do 2 or 3 of those at a time. So these adaptive designs are also called multi-stage trials because the adaptations take place as you go from one stage to the next stage. And you have to pre-specify when those stages are going to occur, okay? And so those are specified stages, and you will have we will go or no go to the adaptation at that stage based on this, or that. And so you have to specify in the protocol that what is going to be the criteria for go or no go, okay? The adaptive design concept is basically thinking in a Bayesian way, okay? And that many people don't realize that that's exactly what you're doing. So we do a lot of things we do the testing of things in a frequentist way, but we really are always thinking in a Bayesian way. I'm going to have some prior information on designing my sample size, but based on what I've observed, I'm going to update it, and I'll have a posterior sample size calculation. That's a Bayesian way of thinking, but people don't think that that's Bayesian. So just keep in mind that these are basically Bayesian thinking about adapting the study. So let's look at some of these adaptations, where they come from. So by definition, adaptive trials are ones that use the data that you've accumulated. So they're using information that you've accumulated to decide how to modify some aspect of the study. But you don't want to undermine the validity of the trial, and you don't want to affect the study integrity. So it becomes tricky sometimes to do so. You may have heard it by different names: flexible study design, or response-driven trial, or dynamic, or self-designing, or multi-stage. Now the latest buzzword is adaptive. Okay? And it requires the trial to be conducted in several stages with access to the accumulating data. Who has access to the data depends on what you're considering adapting, right? And we'll go into those examples. And so just to reiterate again, these are adaptive by design. So you design the adaptation in your protocol. It's not an ad hoc change of the conduct or analysis. So as people say, there's a good quote: adaptation is a design feature. It is not a correction or remedy for poor planning of your protocol. Oh, I didn't plan my sample size correctly, so now I'm going to change it. Oh, I did not do randomization correctly, or the inclusion-exclusion criteria, so I'm going to change it. That kind of stuff. No, it's not, because you did not plan properly. Okay? So, general structure of adaptive designs is that you have these stages, and at any stage, the data are analyzed. You have to pre-specify at what stage you're going to do it. And the next stages may be redesigned, taking into account the available data. So you may have, they call them rules, and you could have during the recruitment, while you're still recruiting people, there's some things that you can change. You can change the eligibility of future subjects. You may have recruited a few people, but now you want to enrich certain types of people into your trial, and those now you're adapting the eligibility. You're basically changing the eligibility criteria, or you decide, you know, these people should not be included here, so now we don't want to include them. We want to exclude these people. We do adaptive allocation, so we can, future recruited subjects will be randomized, maybe not in a 1-to-1 fashion, but in a 2-to-1 fashion. Or we're going to do something different, so we can adapt the allocation while we're recruiting. We can also adapt the sample size. I can not recruit more. I don't need more. I said I needed 500, but I can get away with 400. I will stop it and not recruit more. Or I said I needed 500, but actually now I need 800. So now I will adapt it and increase it. And we'll talk about that sample size re-estimation while you're recruiting. Or you may want to enrich certain subgroups. You decide that, you know, younger men should be included. We are including them already, but I want to give them a higher probability of being included. So my screening is going to go differently. I'm going to do that, etc. And I'll give a quota for the young women and the older men and the older women, that once I get to that number, I'm not going to get more of those, so I will enrich it with this other group, right? Because of special interest or whatever you want to consider. That's during the recruitment stage. You can modify who you're including, how you're randomizing. But after you've recruited everybody there, what can you do? Well, you can still change things if you've put it in your adaptive protocol of sample size. You can still do sample size re-estimation. Yeah, we recruited all the 500 we said we needed, but now we think we need another 400 more. Okay, that's a little bit more problematic to convince the sponsor to give you more money, but if you put it in your protocol and they've approved the protocol, they're willing to do it. What we always do, and this is what we talked about yesterday, is the adaptation of when the trial ends. We programmed the trial to be for 2 years, but now because of ethics, we're looking at interim, and now we need to stop it according to the DSMB recommendation, 4 months early, 8 months early, whatever, because of efficacy, for harm, or futility. We can also adapt other design aspects. So there's many things in your protocol, how you do, you know, some operation, some process, how counseling is done, how patient care is done, the frequency of follow-up visits, what is done at a follow-up visit. I'm not going to take any more questions on the mental state of the person. We're interested in looking at cardiovascular disease, but when we think that the mental status of the person, cognition is affected. And so we want to study 400 people on this. But beyond that, we're not going to do any more of this. So now you're changing, you answer the question on mental health as a sub-question in your study. So now we don't want to do it anymore. So there's all sorts of things in the protocol that you, in sub-studies that you're planning on doing, and you can adapt how much to do for them and when you want to do it. People do all sorts of stuff. They complicate their lives many times to do these sorts of things. So during the conduct stage and during recruitment stage, you can do things. You cannot reallocate differently because you already allocated everybody during the conduct stage, unless you have new people because you've increased the sample size. So what most people find very, very attractive to do is sample size re-estimation. And so we'll go through this slowly. It's also called response-driven reassessment. So it's basically fine-tuning the design parameters in the middle of the trial due to the uncertainty and the assumptions at the planning stage. Because when you are planning the study and you've all done your calculations, you say, oh, the event rate is going to be such and such. The proportion in this arm is this, the proportion in the control arm is that. So I expect to get an effect size of this. Where did you get that information? From some other study that somebody else did in a different population under different conditions. Maybe it doesn't apply exactly to the people you are studying in the settings that you are doing. Maybe that was in urban settings in a high resource country. You're going to be doing this in village hospitals in southern India. Does that same event rate apply? Probably not. So then maybe you want to consider, let's find out early in the study, what am I getting, and do it. And you can do it in two different ways. They call it the blinded sample size re-estimation or the unblinded sample size re-estimation. Blinded means that you're not looking at the rate in one arm versus the other. You're looking at the overall event rate. So, or you could say, if I have a continuous outcome, maybe the variability in the continuous outcomes that I use in the calculation came from another place, and the variability that I have because my instrumentation is different, and I'm looking at weight as my outcome or change in weight, and I cannot afford the Tanita weight scale that is very precise. I have using something else that I'm standardizing and calibrating, but the variance is higher. So I don't know what the variance is, and you didn't do the pilot study to know the variance, but now you find out, let me find out what the variance is, and now I want to recalculate it with a higher or lower variance. Or you have a pooled rate for binary endpoints, the overall event rate, you use that, okay? And if you do it in a blinded fashion, there's no problem with impact in the type 1 error. And regulatory agencies will be okay with you doing this, no problem with that. The only one that's going to have a problem with this is whoever's giving you money. The sponsor may have to agree to say, okay, yeah, I need actually more. And so the sponsor will say, I'm not going to give you any more money, or I will give you more money. And so now you have to convince them that this is okay to do it. But you usually, since you put it in your protocol, you've discussed this already, that you're going to do this potential adaptation. And you will say, I will go from 500 to 800, or I will go from 500 to 1,000. And that has been approved. So, or I may go, I may not go, but at least the maximum will be such and such. And then the company agrees to it, and then that's fine. Unblinded is a little bit more problematic because now you're going to argue, I'm going to do it on the effect size. So I will know how much effect I'm getting, the difference between the two arms. So the treatment effect estimate is calculated at the interim analysis and used for the decision. So who looks at this? The only ones that can look at it are the unblinded statistician and the data safety monitoring board. And if they say you go and increase, then the blinded investigators get a hint. Oh, maybe the effect size is too small, and that's why we're having to increase. Or maybe the effect size is already very big, so we don't have to increase. And people start guessing. So it allows for this interim efficacy or futility stopping, but it's problematic because it is potentially unblinding of things. So it is discouraged to do unblinded sample size re-estimation. We have done sample size re-estimation with the blinded case. And it was one study on atrial fibrillation, and the investigator had gotten prior approval from the sponsor that they would be willing to add a little bit more or not. The way to think about it is, actually, this is a great article on sort of reviews the sample size re-estimation process so that you, again, and it's Bayesian thinking. So I stole this diagram from that paper, but you basically have 5 zones that as you schedule the time. This is for the unblinded one, right? You're doing it at the interim analysis, where the time when you say you're going to do it, and you will see the effect that you have at this point. Are you in the almost ready to show efficacy zone, or are you almost in the zone of showing futility? In either one of those, you will not change your sample size. But if you are in the favorable zone for it, or you are in the unfavorable zone for it, you may have stopped the study there, but if you're in the unfavorable or in the favorable one, you will go up to your minimum sample size. And this is one way of keeping people maybe blinded. It says, we're not going to change the sample size, but you're not telling them if you're favorable or unfavorable. But if you're promising, there's a promising zone that has been prior determined, then they will go on to them. So you have to specify what is your promising zone. And so people do simulations based on conditional power. The same things that Professor J.S. Elan talked about yesterday. What is the conditional power under the null? What is the conditional power under the current trend? What is the conditional power by design? And this is what is done then by that unblinded statistician discussing with the DSMB, and the DSMB will say they won't say whether they're promising, whether they're favorable or unfavorable, but if they say, according to your protocol, yes, you should go to the larger sample size, then everybody knows it's in the promising zone. But if they say no, they don't know if it's favorable or unfavorable. That article gives a nice example where the sample size they show that the sample size is a function of the interim result. And so they had the treatment not efficacious or the treatment efficacious can be unfavorable or favorable. And for a particular final analysis, sample size could be the smaller one or the larger one there. So this is what people do in sample size re-estimation. Yes.
So, my query is, how does the sample size re-estimation in packet fashion work for the event rate? Because when you're not unblinded, but the overall event rate.
Right.
The sample size re-estimation works, and any sample size method works by calculating the number by R.
Right. So you would use the effect that you used in your design. So then you don't change it. So the overall event rate is changing, but the effect, the difference between the intervention and the control is what you put in your protocol. They will not update it with the current effect that you're seeing. With the unblinded one, they're updating it with the observed current difference between the two arms. The other one is using the difference between the arms that you put in the protocol. So that's why you don't unblind people.
But event rate can be very much higher in
Yeah,
one arm
yeah,
as
it
compared to
could
the
be.
other one.
Yes,
But
could
because
be.
you're not unblinding and you're keeping the effect size the same for the re-estimation,
It's
so
not a great thing to do, but it doesn't penalize you for, doing an interim analysis. Here you're doing an interim analysis as well. Yeah. Mhm. Here you're changing the difference here. Is it proportional?
It's the sample size that we are recalculating. Your primary outcome variable is suppose it's a continuous. As you said, we can do the blinded one because we can change the sigma with the variability. But
if you're looking at variance, but if you're looking at effect,
probabilities, how would we like do it?
No, but you would be, so you would have had what is the mean in one arm, what is the mean in the other, because it's continuous. You'll be looking
the
at a
continuous
difference.
is fine, sir. But
Yeah.
if you are looking at probabilities,
Oh, if you're looking at proportions, okay.
proportions,
If you're looking
fine.
at proportions, you would have to say, what is the overall proportion? If I put the two arms together, the event rate is 40%. But you put in your calculations, originally calculations, you would have put 10% for one, and well, let's write up some numbers here. So you have the control and you have the intervention. And when you're designing the study, you would have had some event rate for this. Let's say this is 10%, and you expect that you will get better cure here. So you expect this to get 15%. What is the overall? Overall is 12.5, right? Now in the blinded one, what is being presented is that, oh, wait a second, we have 8% here. They don't see this. They don't know what's happening here. They will guess that this is, 0.5 or 50% higher than this because they know this. This is the design. So they will say, oh, if I see 8%, maybe this is 4% and this is 6%. Or maybe this is, 5%. And so therefore this will be 7.5%. But that cannot be because this is 8%. It has to be a little bit higher. Sorry. So that this one here may be 6%, and then this one would be 9%, so that the overall is 8, but they don't know this. This is in the blinded one. In the unblinded one, they will say they will be given the 8%, and they will be given this and this. But when you do this, what he's asking, he says, how do I do the calculation if I don't know this? I say, well, assume 1.5 is the relative risk because that's what you use. You had a relative risk of 1.5. So now assume a relative risk of 1.5, make up. So if this is 6%, then 1.5 would make that 9%. If this is 7%, then 1.5 would make that 10.5%. Oh yeah, sample size will be very different because here the difference is 3. The difference here is 3.5. But you don't know. Yeah.
So supposing the interim analysis says the event rate is 8%, the ratio is 1.5. Therefore, I keep, if this is 8.5% in one arm, it should be 12 in the other arm.
Mhm.
That is how I decide. Is
Yeah.
that right?
Yeah.
Difference is same.
So if you're
She's-
working on differences or you're working on ratios, right? That's you don't know. If you're working on differences, you say, oh, the difference here
It's 5%.
was not is 5%.
This
So
is
then here
8%.
you say if it's 7 here, then it is 12% here.
So how do you get that 7%? This is the interim data, overall data.
Yeah. Right. So in the blinded one, you don't. The blinded one, you have no idea what that is. Yeah.
To smaller extent.
Smaller amount, right?
Smaller extent.
So that's why we don't like ratios, but people like ratios. But what he's saying is that we will go with that 5% because I will say if it is this one is 6, this is going to be 11. If this is 7, then this is going to be 12. If it's 8, this is going to be 13. But doesn't these don't make sense? If the overall is 8, you have to be overlapping. So, but when you do the formula, the difference here is 5, the difference here is 5. You are guessing. No, you cannot do it in the blinded one. But it doesn't matter. It doesn't matter because the difference is always 5%. So it's not going to be different. It's
different, no? It'll be different, right? So the difference is we keep as 5, but whether P1 is 6 or 7 or 8, the numerator will vary. So if you have a table where you have a range of values, because we have computer, we can do that, no? So what are the possible values, and then we take the maximum sample size based on that. So
but all the values will be the same.
that's what I was asking earlier, sir. If the P1 and P2 changes, will the sample size
Let
change?
me explain this. This 8% comes from your interim analysis data, overall event.
Yes, sir.
Yes. Now we need to decide what is your P1 and P2. What you know is from the already given data, it is 1.5%. The ratio is 1.5. So from that, we need to decide what should be the P1 and P2. Supposing you are saying this is 6 and this is 9. You're saying if you put 7 and 10, it is going to be different. These are the small difference.
Small
versus because it will not go wrong unless it is 2%.
And it depends on what you think. That you're thinking in terms of differences, those differences you will use the same. But if I'm doing it, I'm guessing what they are. And if they're different, yes, it will be different. You can put a table if you want to, but you have already put it in the protocol. Is that whatever the event rate is here, I will assume that if I get the interim, this is the interim, overall rate is 8%, I will assume that it is 6 and 9. Or what? That's it. You put this in the protocol. This is in the protocol. And then you have the sample size for that. And that sample size will require, here you had a 5% difference. Now you have a 3% difference. And so your sample size will go up. And you will say, here I required originally 500. Now this will make me get 1,200. So please give me 700, money for 700 more. But this is in the protocol. This part is all in the protocol. If it's 8%. And you say, oh, if it is 9%, then I will assume this. And that will require this. And then will require that. And if it's 7%, I will require this. I will guess this, and that will require this, and this will get more. So you put that in your proposal to the sponsor, not in the protocol necessarily. You could put it in the protocol as an appendix, but the main thing is all of these sample size re-estimation, you are going to ask the sponsor to give you more money if you have to increase it. I also advise you that this is what you had planned, that if you now do, what if I have 13%? I'm going to assume such and such. I will not require 500, I will require 450. And therefore, we will not recruit everybody. We will give you money back, Mr. Sponsor. That kind of stuff. Be nice to them. Don't always ask for more and more and more, because then they will like it and then they will fund you. Otherwise, they may not fund you to do it. Okay. Oh.
Sir, I understood in unblinded how we can sort of find the intervention and the control arm, the effect size.
Mhm.
But in the blinded arm, my doubt is, sir, what is the harm with?
This was the blinded. What we did was blinded.
What we did is blinded.
Yeah.
In the unblinded, I understand it. In the
Okay.
blinded, what is the harm with assuming that the control, because you already start off with data which is not from your study, but you at least have some data to suggest it maybe 10%. What is the harm with, because you're assuming two variables now for one outcome? What's the harm with if it is 8%, you at least start off with 10% because you already have data saying it may be 10%. Then you have only one variable which you're assuming.
You can assume whatever you want to, but if you say that this is 10%, because that's what you had from previous information, then to be 8%, this has to be small. So this is doing worse.
Yes.
Right? Okay,
But that's
you can
possible.
do that if you want to, but you don't know. Yeah, if you want to do it, you can do that, and then you may tell the sponsor, we are actually probably in the futility zone, and so, or unfavorable, so we're not going to ask for more. So you can put all of those scenarios in your proposal, that if we have this, I will trust that this is 10. So therefore, I expect that we are actually not doing well. We may not want to recruit more because we are likely to be in the unfavorable or futility, and will be done like that. Yeah, that's possible.
So because this is just a hypothetical situation, is there any way of knowing actually based on whatever adaptive trials have already been done, which form of assumptions actually are accurate or?
So yes and no, because people don't publish this stuff. Right? They don't tell you. If they did an adaptive, they will maybe tell you that they increased the sample size at stage 1 to stage 2 because you met the criteria, but they won't even tell you their criteria, right? Because they don't think that that's important to give proprietary information, because now you may not hire me to do the work for you or whatever. So people don't tell you their secrets. So that's tough. But you could write to them and ask them. They won't tell you, but you could ask. Okay. But this is very commonly done. So sample size re-estimation is a very attractive thing. The danger is not with the blinded one. The danger is with the unblinded one. So in the blinded one, so you have that there. In the unblinded one, they are told, the DSMB sees, they see 8, and they see, let's say this is 6, and this is 10%. And so they see this. And then they will say that you will also have the same thing of favorable, unfavorable, and it looks very favorable, and they tell you to stop recruiting, or you don't need more people because you're already very favorable. And if you only have it to not increase with the favorable, then you're unblinding. So that's why they have favorable and unfavorable, so people don't know which direction. So it's still possible to do it, but the message of the Data and Safety Monitoring Board as a feedback to the investigators will say, we carefully looked at all the data presented to us, and we do not see any reason why you should change your sample size as per protocol. They don't tell you whether it's favorable or it's unfavorable, but they have to be careful in how they say it. They can't say, oh, it's looking good, so you don't need to do more. They can't say that. Or it's looking poorly, you don't need to do more. You don't want to unblind it. All right. Yes.
What would be the message to the researcher believe it is promising from the DSM?
They basically would say, following the recommendation, following the protocol. So we've carefully examined the data presented to us, and according to the protocol, we recommend that you follow the increase in sample size. That's it. Well, they're in the promising zone, yeah. So it isn't. It's sort of unblinding, and that is the bad part of the whole thing, right? But they could be. The promising zone is, some of it is unfavorable, some of it is favorable. So we don't know, but it is every adaptation is potentially unblinding. So while adaptive designs are attractive, always keep in mind that everything from the interim analysis adaptation to the sample size re-estimation, and you'll see now for the eligibility criteria, the enrichment ones, all of them are potentially unblinding, because you're changing something based on what you've observed. And so if you change it, they know what you observed, or they can guess what you've observed. If you don't change it, they can also guess what you did not observe. So doing adaptive trials are very, very tricky. They seem very attractive, but they're very tricky. And so you have to be very careful not to unblind. Adaptive enrichment is one of those also pretty common ones. And that's, again, using interim data to identify which types of patient subgroups do better or not do so well. So that is very much unblinding. And so you say, wait a second, the older people are not doing so well, so I don't want to necessarily get more older people in this. They argue it from an ethical standpoint, right? Because why should we get people that are not benefiting from this to subject them to be in an intervention? So they enrich the sample sizes of the people that are doing well or within subgroups. And this is, again, another Bayesian thinking. So they will decide that, okay, I need to get this particular subgroup more of these groups. So they'll have different types of subgroups, and it'll say, no, okay, we'll just keep the blue and purple ones, but we'll get more of the yellow, orange ones to come into the trial. Okay. So now they're saying these people are the ones that are benefiting more. So that at least it's communicating information that somebody is benefiting more and some people are benefiting less. So it's some unblinding, but not the differential between the intervention arms. Okay. So they call this outcome adaptive randomization sometimes. Okay. But it should be more quota. And what is outcome adaptive quota allocation or something like that. Okay. Yeah. So let you not stand here. That's a good question, because the sample size will be still the same, and these subgroups are basically strata. And if they think that a stratum is going to be of more benefit, they should have probably said in the protocol we are going to, we want to have adequate power in that, because they will end up, if they enrich in one arm and one subgroup, they'll have more of that people, they'll have power in the orange, but they will maybe not have now power in the other ones. But most of the times, the subgroups, in a case like this, you are correct that they are interested in knowing the effect in a particular subgroup. So they better power for all subgroups, because it could have turned out that the blue was doing better. So now I need to have power in that one. But so I need to have the sample size. So this is a little problematic if I want it, because when we do the stratification, we do it to balance things out, but not necessarily to test it in each one. But when they're doing this, they're thinking, I'm going to be wanting to test it in the orange one, or it could have been in the blue one, or it could have been in the purple one. So then they have to have adequate power in all of that. It's a good question.
Selection bias, because we are changing later on everything, the subgroups also.
No, it will maybe change the external validity, who I can generalize to, because I will be able to generalize to more orange people than to blue and purple, but not bias, because the randomization, I'm still randomizing equally in equal proportion to intervention or control. So this is enrichment. The other thing that is problematic is the next one, which is adaptive allocation. And this is when they say, oh, the treatment is working better in a group, so, or is working better. And so I would like to give more people the benefit of getting the treatment that's working better. So I want to now, instead of doing a 1-to-1 randomization, I'll do a 2-to-1 randomization. So you have 2/3 chance of being sent to intervention versus 1/3 chance of being sent to control, and treatment is doing better, so I want to do this. This is very unblinded, very unblinded, because now that they know that this is doing better, that's why you're doing it. But people even consider this as an adaptation. Why? Because it may help your recruitment. Because you could, if people say you have a 50-50 chance of getting the intervention, most people make up in their mind that they don't know, they don't understand equipoise. They think that I always want to get the new intervention versus the standard, because they always assume that this new is better, whether it is or not. But now that you're doing this and saying, we're going to do it 2-to-1, even people will say, oh, I have a better chance of getting the new one. And it must be because it's working better. Now you've unblinded almost everybody. Participants.
Revise the consent form.
Yeah. You have to change the consent form.
Let us say is not responding well to a certain intervention or SOC, standard of care, and we decide to hence sort of, how should I put it, maybe I don't know how it is done, but essentially we maybe recruit fewer of that age group. But there is a chance, since it's not powered for that, that the consequent or the later participants who would have received the intervention may have responded better. So this is difficult for me to swallow, because it seems as if it seems premature to make this decision. The whole purpose of the sample size is to prevent these kinds of things from happening. There is intra or inter-individual variation in the responses.
So one correction is that you're not doing it because you're observing this. You're putting it in your protocol. We are not sure if elderly people are going to benefit that much. We will look at the elderly after we have 100 elderly. And if it's doing well in the elderly, we will continue this. If it's not doing well in the elderly, we will not recruit more in the elderly. So you put it in because you have thought about this before, not because you happen to say if this group is doing better, that group is not. you have biological reasoning that
Okay, so
argues that I want to
so
put it
that's
in the
somewhat fairer. However, the number which we redefine in our protocol, that should be something which gives us adequate power to say it with some conviction that yes, it does not seem to be showing much effect in elderly, and hence we will reduce the recruitment. I mean, the sample size should ideally change or increase because of this.
Yeah, and so you may say that if I get rid of the 100 elderly or put a quota on it, and I will only get 200 elderly, and I will then have the overall sample size will be, let's say it was 2,000, and I was going to do 100, however many elderly, but I would still get a total of 2,000. Now I will only get 200 elderly, and I'll get 1,800 non-elderly. You have to have calculated, do I have power in the non-elderly with 1,800 to answer the question, or we're still going to have to do per intention to treat all 2,000, right? But it is unblinding because now you say, well, there is a group in which it's not working. So there is some information there. Again, I tell you, these are things that people consider. No, I'm not forcing you to do it or want to do it. There are people that argue, some, people that have mild disease, it's not going to work so well in the mild disease as in people that have severe disease. But for ethical considerations, I'm going to have to include everyone. But I'm going to not want to go on. I'll have quotas. I'll say we have this much quota for this, for this, for this. And if stratified, basically, and if I will change the probability, change the selection criteria and change the quota so that I will only get so many people of the blue, I will only get so many people of the purple, so many people of the orange. And they will say, again, favorable or unfavorable. Maybe I've answered it already. It works well in the blue, so I don't need more blue. And it doesn't work so well in the purple, so I don't need more purple. But it works this. So they try to do it so it doesn't unblind. But basically, they're enriching or changing the proportional distribution of different strata.
Right. So, what is your personal opinion on adaptive enrichment and adaptive?
I think it's just too much tweaking of stuff. Basically, my personal opinion. People are very much attracted to the fact that, oh, I can change things. They don't know how much trouble this causes. They don't understand how much care has to be done to keep people unblinded. Blinded. Keep people blinded as to what's going on. And the, interim analysis, you heard how Kumar has to hide so that he doesn't unblind investigators. When you're doing these things on a daily basis, this is not at an interim analysis. That's a stage is coming, and now the investigator comes to your office and says, "What's going on? Are we in the promising zone? Are we doing this? Are we doing that?" You go work from home. People harass you asking you these things because they want to get unblinded. Every investigator I know does not like to be blinded. They say they will do this, but they don't like it. And so they try to get unblinded. And so these things, they find it attractive also because they can tweak things and it also helps them guess what's going on. I don't like them personally, but people do it. People do it. So I'm just telling you that that these things exist, right? So sometimes yes, sometimes no. Sometimes if, like, for example, if you're doing the blinded sample size reallocation, then that is the steering committee can decide. You don't need the DSMB to tell you that because you have in your protocol what criteria will make me go, what criteria will say don't go for that. And so you don't, if it's unblinded information that's needed for the in part of the criteria for the change, then only the DSMB can do it. And if it's based on blinded information, then the steering committee can do it.
I can change the number of
Oh, yeah. So the study protocol will be very detailed. As a stage one goes from this number of patients, this number of patients, or this number of events, this number of events. When we get to the end of stage one, we will do this. We will do this calculation. If we get this value, then we will do this. If we get this value, then we will do that. If we get this other value, then we will do that. It's all detailed in the protocol.
So then how they're concluding this? If their outcome is composite measures, and in that composite measures, I'm taking skin, joints, and in between the study period, I'm just taking people who was one of my drugs is responding to joints, and I'm taking after few period only joint patients. And at the end, the study is positive because composite everything is positive. So how they are concluding at the end with such a design?
I mean, if you have your criteria involves multiple outcomes, then you specify that in the protocol. At the end of stage one, we will look at the skin problems, and if it's this or this, we're going to do that. We will look at the joint problems, if it's this or this, we're going to do that. If it's skin and joint, we're going to do this and this and that. If not, we're going to do that. We're not going to increase the sample size. We'll enrich more of these. We'll do less of that. We'll change the allocation. All of that you have to think about and put it in your protocol, what you're going to do at the end of stage one. And many people do stage one, stage two, stage three. They love to complicate their lives. Do that. But if you're doing adaptive, you have to say what you're going to do. And if I have the joint one this way, and I have the skin this way, then I will do that. If I have the joint this way, but not the skin this way, then I will do that, etc. All of these things have to be pre-specified in detail in your protocol. We call them adaptive.
Adaptive randomization, you have to predict the outcome, right? It's treatment outcome that we talk.
Right.
So what if there's a treatment response which is not being predicted? If you get a response which is not being pre-predicted, where do you adapt them? Like you should have room for.
Well, so that is not an adaptive trial.
Okay.
That is now an ad hoc change to the protocol because maybe it's a severe unsuspected adverse event, SUSAR that came up, or something that you have not anticipated is not an adaptive design. It is now maybe requires a change in protocol. We had this in one study where I don't remember how to lobar ICH, something with the stroke folks. You know better than this, but they had not thought about people with lobar ICH were included in this trial. They are particular higher risk for problems, and they were getting a lot of adverse events on this. And the Data Safety Monitoring Board said you should not. This is unethical to keep these people in the trial. This was never thought out before. It's not in the protocol, but they changed the protocol. These people are no longer eligible. They should not be randomized. You can change it, but it's not adaptive. That's the key point I want to make. Adaptive designs are designs that have the adaptation at the beginning in the protocol, not things that you change. You still can change lots of things, but those are changes to the protocol, amendments. Sometimes they require the IRB to look at it. Sometimes you have to get the the sponsor to agree to it. You have to get this participants to agree to it, whatever. But you have, those are different from adaptive designs, okay?
In an adaptive trial, because you are proceeding, you don't have to go back to the IRB in between.
Right, because the IRB has already looked at the protocol and
They
approved.
know, okay.
So they know the adaptations you're going to do.
Have you implemented it?
No.
Okay.
I don't like to do it. And most of, we don't do, a lot of these things are done maybe in phase 2 trials more than in phase 3. Our small our trials are small trials that we do are around 5,000 people. Normal trials that we do are around 12,000 people. That Compass trial we did was 28,000 people. And the main thing that we've actually done is sample size re-estimation, trying to bring it down sometimes because we're doing these large trials. But all of this other stuff, it's attractive. People think we can do this, but it's a pain to try to do this kind of stuff. Operationalizing and trying to keep the study blinded is another issue, right? You don't want to do these things because they're potentially unblinded. Yeah, we'll get into that. All right, so adaptive allocation is problematic. I think it's this one. Adaptive allocation is problematic because it is very potentially unblinding. If you say now we're going to do 2-to-1, we're favoring the intervention, this one, because it's doing well, usually people would guess. Here's an example of a response adaptive randomized double-blinded trial done in India at AIMS in Bhubaneswar. So they were patients of low socioeconomic status undergoing cancer chemotherapy who cannot afford this NK1 receptor antagonist to prevent nausea and vomiting from the chemotherapy. So they had this thing called olanzapine plus pregabalin orally for 5 days as an add-on to standard antiemetic therapy, and the placebo orally to emulate the other one as an add-on to standard antiemetic therapy. And it's the proportion of patients with overall no nausea. And it was 5 days only. And they did this interim analysis where they actually changed then the allocation and gave more people the olanzapine pregabalin one because it was doing better. But they got this done, and they don't give details of who made the decision and whether it potentially unblinded people or not. They just reported that we did a response adaptive randomized double-blind study. I don't know how you adapted. I would have to go back and read their protocol, which I have not done. But people do this kind of stuff, so don't think that people don't do it. So what they did was the original sample size was 42 and 42. They needed 84. And so they said, we're going to do the interim at 15 and 15. So the first 30 patients is when we're going to do it. And the second stage, we will decide to favor the intervention or not according to that interim look. So then at the interim look, they said, somebody said, yes, unblinded, intervention is doing better. And now these are, because what? These are cancer patients. And it's a life or death for them, right? So they would prefer to be getting the intervention if it's working. So then the next 54 patients were randomized 2-to-1. So very much people know that it's being unblinded, right? But the intervention was blinded. And so they did not tell, the way they kept it secret was they didn't tell anybody. They told the statistician, you just need to change the randomization scheme. Or no, they didn't say this, but my guess is that originally in the protocol, when they designed the study, they had 2 randomization schemes developed. One that was 1-to-1, and one was that 1-to-1 up to 30, and then 2-to-1 for the next 54. And the unblinded statistician is given access to go into that computer and say, switch from this randomization scheme to this one. Nobody knows. And if the interventions are blinded, they're getting this, whatever, they're getting that additional olanzapine and pregabalin, but the other ones are getting placebo. Nobody can tell if it's truly blinded, it remains blinded. And so they ended up with 51 versus 33. So it has to be tricky to do it, but it can be done, right? And so if you think that, but what do you tell the patients at the beginning of the trial? You say, we are going to do informed consent. You have to be transparent. We are going to be randomizing you to this new treatment versus the standard of care. And you will have, we in general will be favoring the new treatment if it's doing better, and you'll have a higher probability of getting it. They won't tell you the probability. As long as the IRB lets you get away with that, you can do that. They won't tell them the details after the first 15 people or 30 people will have 1-to-1, and then the other ones may or may not have 1-to-1 or 2-to-1. You don't have to give all of that information, but you have to do in the paper, they give that information.
Situations like this, when you're doing initial sample size calculation, the assumption is with so much power and so much alpha, this much is needed in each arm to answer the question. Now, in this situation,
You have to do two calculations.
okay.
You'll do two calculations so that if this one is doing better, how much better is going to be the trigger for you to change the allocation from 1-to-1 to 2-to-1. And then if it's that much better, that's the effect size that I'll use for the next 54. And then calculations become a little bit more cumbersome. It's not any software that's easily available. You will have to have the statistician program it to then do simulations to get what is the power. If I do this much this way and the effect is that, and you change, or the effect is this and you change, or the effect is this and we did not change. So they have to do all of these calculations before the protocol is finalized.
So when the final reporting comes, the power and alpha, is it like it has to be maintained throughout the study with whichever final calculation they get, or is it possible that there may be a power when they started and a different power once they do the adaptation?
So the power will be changed when you do the adaptation, because that's exactly what you are expecting. That's why you're doing the adaptation. You're doing well in one arm, huh? And so you have a big difference and you'll have higher power. But at the end of the study, power doesn't exist, right? Remember, and I was talking to somebody this morning, power is a probability, right? This is what Seelan mentioned. Power is the probability of committing 1 minus the probability of committing the type 2 error. Probabilities are always in the future. They're never in the past. You don't have the probability in the past. Just like the example of rain. We talk about the probability that it's going to rain today is 7%. Nobody says the probability of it going to rain yesterday, because yesterday it either rained or it did not rain. There's no probability. And so there's certainty of what happened. And so the same thing, at the end of the trial, you either reject the null hypothesis or you don't. And whether you reject or don't, you don't have any power. You just have what your action was. You reject or not, it's like it rained or did not. But if you wanted to, people like to calculate what would have been my power if I had seen this and I had this sample size. Now that's a theoretical exercise, but it's not really useful or meaningful to do, but a lot of people do it. And that's speculation. And that's, I can tell my wife, why did you tell me I should have turned back there when I'm already past it? And I cannot make a left turn now. It's already in the past. Don't tell me these things. Tell me before I get there, so that I know what to do. Okay, all right. Well, let's go on because we have a lot more on these adaptive designs. So people do other things, right? So we now have these sequential multiple assignment randomized trials. They're called SMART trials. And this is actually, some people call it individualized medicine. They call it precision medicine, but it is taking multiple stages of randomization in a patient. So it's not in a group, it's in a patient, so it's individualized. So treatment assignments can be adapted based on patient responses. So now it's not outcome response, it's the patient, how they are doing. So it allows researchers to evaluate different treatment sequences. It's almost sort of like a platform, but the platform now is the patient. It's not the infrastructure that we had before when we talked about platform. And I think Ananta mentioned this also, that you can do platform trials, and this is essentially a platform trial in a given person. And the person serves as their own control. Okay. There's no randomization. So for me, these are quasi-experimental designs. You're not randomizing them, so that you are assigning new treatments or not to them. But you evaluate different treatment sequences and identify the most effective strategies for an individual. And if you have multiple individuals, then maybe you can do it in aggregate later on, but you've treated each one very differently, okay? And it becomes tricky then to know what works overall, because then you have to worry of washout effects, just like in crossovers. You have to the washout effect, the latent effects of one or the other, all of these things affect it. So it's very different type of design than your standard design. But the key features of this is that they're sequential. They are adaptive. So what you're adapting is the intervention that you give to that person, okay? And it's based on how they're responding. So you can tweak it. So it's basically, folks like to do this because this is how physicians actually treat patients. Patient comes to you with a problem, you give them this regimen to follow. If they're not doing well, you give them this other one. And if they're not doing well, you're doing another one. So this is going and doing different things. And what is the re-randomization? Why do I say randomization? It's because you have 4 or 5 different things that you could give them. And now you randomize which of those you're going to give to that person as the next one, because this one failed. And then you get the optimal adaptive interventions by, now it's more complex analysis to get that. And they are trying to, as they say, bridge the gap between the artificial nature of clinical trials to the real nature of treating patients, where you are sort of tweaking things and going back and forth, et cetera, real world clinical practice. So their personalized treatment, maybe you're more efficient, I don't know. And the patients certainly should benefit from it. But understanding which is the best of those treatments, now actually it's done with Bayesian methods. I should have put that there. There's a software called WinBugs. WinBugs will say now we can rank which of these treatments gave us the optimal result. This is what they use in network meta-analysis. And maybe you'll talk about the Bayesian ranking of the different interventions when we do network meta-analysis, very similar to what we're doing here. So you'll do Bayesian analysis for smart designs. The final 2 things I want to talk about of adaptations is, we don't think about it this way, but when we start the study can be adapted as well. We think about it as fixed. Yeah, I write my proposal, I get the money, I have a planning stage, bam, I start the study. Okay. But I can start the study earlier or later, and I can decide when to start my study. And so doing a pilot study is very different from doing what we call a vanguard stage. Some people call it internal pilot. So it says I start my study now early and decide to start it early before I have actually finalized everything in the protocol. Why would I do that? Because I still need to refine some of the processes of how I'm doing recruitment, or some of the processes of how I am setting up the lab, and how am I doing this? But also I want to, we do it a lot because we haven't gotten funded yet to do the study. And we have seed money that we get in our organization for a novel idea. Why don't you start recruiting people under this protocol? Don't change the protocol, but show that you can do it. And once you have some preliminary results of recruitment, of retention, not outcomes, but of process, now we go to the sponsor and say, see, we can do this. Give us money for the big study. And so we do this. We started a little bit late to do it. So the main trial, I mean, there's uncertainty about feasibility. And so this is really a feasibility study, but it's not a separate pilot, because if you do a separate pilot, those patients cannot be used in the analysis of the main trial. So this internal pilot, then the patients are part of the main trial. And that's sort of the benefit of doing it by doing this early startup. Okay. And you may change a little bit about the sample size because you get some more information, but you're not changing the actual protocol of the interventions, how they're administered, et cetera, or who is eligible, who is not eligible. You're not changing any of those things. So that's one thing that people are doing sometimes now as starting. We saw yesterday, and I'm not going to go over this again, that interim analysis for possible early termination is an adaptive design. And we have that in our protocol. We are going to do interim analyses. And this is how we're going to do it. And then we have a separate, very detailed DSMB charter that has a statistical analysis plan for how we're going to look at futility, at safety, at efficacy. And we have go, no go guidelines. They're not rules. They're guidelines that we give to the board to decide yes or no, this or that, etc. And they can be based on the group sequential, or they can be based on conditional power. Sometimes it's also, they will look at it and say, the event rate is low. Maybe we should extend to get more events. And we can do power based on the number of events, not only on the effect size as well. So people do these sorts of things. It's considered then an adaptive design because you're doing adaptive ending of this trial. Yeah. So you don't decide it. Your data safety monitoring board decides when they want to do it. Yesterday, Kumar said that at PHRI, we usually don't do it until 50%. But not everybody does that. Anybody can do, any board can decide when they want to do it. Most boards are reasonable and they say we want to have robust amount of information before we start doing that. Because it's that alpha spending function approach. I'm not going to start spending my alpha until I'm not going to start buying anything until I have decent quality stuff to look at. So they don't have decent information early on. Overall interim analysis, not by arm. What is that?
They're not unblinded, but they have the entire, all the enrolled participants' data. And so on that, you said the data analysis can be done by the research team.
For what? I didn't say that.
There were two types of interim analysis. One by the DSMB, that's fine.
Okay.
They're privy to all the information.
Oh, okay. So yes, people call it interim analysis, but the unblinded interim analysis is the interim analysis. The blinded interim analysis is monitoring the study, the monitoring the study progress, etc. But some people call it interim analysis. You're right. Okay, so for monitoring, when you started is day one, from first patient or from screening. When you start screening, you start monitoring how is the screening process going. You start monitoring how is the recruitment going. As soon as the first patient is randomized, you start monitoring adverse events, overall adverse events. You start monitoring this. You start monitoring from day one. So yeah, so let's separate out monitoring from interim analysis. Interim analysis is the unblinded look to see whether you are going to potentially stop the study early due to futility, safety, or efficacy. Right? That's different from monitoring. Yeah.
As a part of adaptive designs, depending on how we are
Methodology of recruiting. So yes, that it's not considered adaptive, it's just considered the way we behave, because all we're doing is, I decide I'm going to send mass media, Twitter, or I'm going to put announcements in the buses, or I'm going to call my colleagues. That doesn't matter how you do it. Does that? No.
like we had another study where we were doing first time thing. We wanted to recruit them on an online platform. Thinking that they would come out more openly on the online platform, but coming physically,
Yeah.
and getting recruited and using that intervention. But what we realized after 3 months, online only recruit 9 participants. Not many people were coming in for various reasons.
Right.
So then we shifted our entire methodology to the physical platform. Rather than online, we will now do it like let the people come to the center and get it done.
So yeah, that's fine. People try everything possible, and there's nothing wrong with it. It's not an adaptation. It's not. It's just part of your manual of operations. It's not part of your protocol. In your protocol, you say we're going to recruit. We will be recruiting from multiple sources. That's about it. And sometimes you will say what you're going to try to do, but if it's difficult to do it in the way you try, you will try other ways. And it's perfectly right. In the manual of operations, you will have details of how we're going to do it with the online platform, how we're going to do it this way. Will we do it this way or that way? How do you actually do it is in your manual of operations. You can change your manual of operations. It's not a protocol change to change that, right? And it's not an adaptation either. No. And any way you do it, you're always getting a garbage sample, right? There's a convenience sample. And so as long as you don't change the randomization, that's the difference, right? Okay. Now, changes to the protocol, sometimes you may say, I am going to look at the effectiveness of doing an online platform for recruitment, but after the first 2 months, if I'm not getting such and such, I'm going to consider paying for people to come in and investing the money for this. And so you may have put that in your protocol if you wanted to. If you don't have to, but some people do. And so, but changes to the protocol or design sometimes may come up. Those are change the target treatment difference or the effect size, changing the primary outcome or changing the primary analysis. These are big no things to do. They are frowned upon. You should not be doing that. It should plan properly your protocol, because this is not well seen to change your protocol midstream. So there isn't really a adaptive design for changes to the protocol. Most changes to the protocol occur because you find things are not going well with this protocol, or this protocol has some mistakes, so therefore we're going to change it. And as I said, that's not adaptive design. That is a change to the protocol, a change to procedures, a change to things. And you just have to document that. And hopefully you're transparent when you report that this is what we did. Okay. But that's not adaptive design. A good paper that came out as a review article of adaptive designs in the New England Journal. I thought I had the date on it. I don't see it. But it was 2020, I think it was. All right. We've talked about master protocol, so I'm not going to go over it. So Ananta talked about basket trials, umbrella trials, platform trials. You'll